All right, so welcome back to EBE from your long vacation there of 2 days. And we're going to continue on with photosynthesis. The last thing we talked about was the equation for photosynthesis. And we talked a little bit about light and then introduced the idea of pigment molecules that interact differentially with light by absorbing and reflecting different wavelengths. Right now what we're going to do is ask the question of whether pigments can be photosynthetic. That is, are the pigments found within plants somehow involved in this photosynthesis process? Pretty simple question to ask. To answer that, we're going to go through a two-step process. The first part of this is to figure out whether plants have pigments, and the next step is to figure out whether they're involved in this photosynthesis process. So, to do this, pretty simple little lab we're going to do. The first thing we're going to do is collect a bunch of spinach leaves. And we're going to take these spinach leaves and we're going to grind them up in a mortar and pestle. Right? I'd label them, but I'm never quite sure which is the pestle and which is the mortar. But the idea is we're going to grind this spinach up, and at the end of this process, what we should end up with is this kind of green goo at the bottom of the pestle, like we made spinach pesto, right? We just kind of ground it up into this mushy stuff. Okay? All right, then what we're going to do is take a piece of chromatography paper. I imagine you've done some chromatography in organic. If you haven't, you'll probably do it in the future. You can take organic 1 or organic 2. And what we're going to do with this green goo is with a little paintbrush, we're going to paint a line of this green goo here onto the chromatography paper. Then we're going to take the chromatography paper and put it into a little chromatography apparatus, usually a tube with a screw cap so that whatever solvents we use don't volatilize. And then we're going to put these solvents here in the liquid form at the bottom of the chromatography apparatus, but we're not going to put as much liquid as up to the line. We're just going to put enough here to cover the bottom of this chromatography paper, which will be touching the bottom of the apparatus. Okay? Now the solvents we're going to use are pretty common solvents. One of them is acetone, and the other one is petroleum ether. More on those in a second, but we're going to mix the two, and that's the liquid that's found at the bottom of our chromatography tube. Okay? Now, the thing about acetone is photosynthetic pigments will dissolve in acetone. So if I just put acetone in here, the acetone would wick up the paper, the pigment on the paper would pull off, it would dissolve into the acetone, and then I would just have a bunch of green liquid. So that wouldn't work very well for chromatography because none of it would stay on the paper. The petroleum ether is capable of grabbing the pigments and holding it onto the paper. So what's going to happen is the combination of the acetone and PET ether releases enough of it from the paper so that it can move without it becoming completely solubilized into the acetone. So it's a neat little trick. And then as the liquid moves up the paper, comes up the paper, it grabs that green goo and begins to carry it up through the paper. But if there are different molecules that were in this green goo, it's possible that these different molecules can move different distances up the chromatography paper. That is, some of these molecules will move faster up the paper and some of the molecules will move slower. Now the speed with which this stuff moves is based on, number one, how well it solubilizes in this acetone PET ether. If a molecule solubilizes pretty well, it can be carried up the paper faster, whereas if it's still trying to hang on to the paper at the same time it's being moved, it will move more slowly. And then the other thing is the mass of the pigment. More massive pigments would move more slowly, lighter pigments would move more quickly. So a molecule that's low weight, high solubility would be up here, low solubility, big mass would be down here. So the whole point of chromatography is to separate molecules out, and that's what's been done on this piece of chromatography paper here. There's the loading line, that's where we painted the pigment, and then here there are 1, 2, 3, 4 bands. Okay, for us, we're just going to pay attention to 3 bands. The first band here is called chlorophyll B. The second band in here is chlorophyll A. And the third band is the xanthophyll band. And the fourth band is carotene or the carotenoids. For us, we're not going to talk about the xanthophylls. They're not involved in photosynthesis, and we'd have to do a little bit of chemistry to talk about exactly what they do. So let's limit our discussion to chlorophyll B, chlorophyll A, and the carotenoids. Okay? Now notice there are slightly different colors here. Chlorophyll B is more of a, they call it a Kelly green sometimes. Chlorophyll A is more of a bluish green, and then the carotenoids collectively are any pigments that are red, orange, or yellow. So green, green, blue, and this one's, depending on the particular pigment you've got, it's either red, orange, or yellow. Okay? Now, what let's do then with our piece of chromatography paper that has these bands on it, let's take a pair of scissors here and let's cut each of these bands away from each other. Okay? Let's take this band, for example, that's the carotenoid band, and let's take that piece of paper and let's put it into acetone. I keep wanting to misspell it. Let's put it into some acetone in this thing here called a cuvette. Now, acetone solubilizes the carotenoid pigments, so they're just going to go right into the liquid. So I should have a liquid here that's the same color as the band. Okay? Then I'm going to take this cuvette and I'm going to put it into a spectrophotometer. Photometer. Again, something you should have used in basic biology, right, MCB, also I hope in organic chemistry. Right? Then, let's take a little light source here. Right? That's my little light source. It's giving off light that will pass through the liquid, and then it hits this thing here, which is a photographic plate which is capable of quantifying the number of photons that struck the plate. So the idea is I release a certain number of photons, I count how many got to the other side, and that percentage would be the transmitted light, and then the stuff that didn't get through here is referred to as the absorbed light. So typically what we're reporting is percent absorbance. Okay? All right, now, the acetone itself absorbs and transmits light, different wavelengths. So typically what we're going to do is take some acetone all by itself. We'll refer to this as the blank. We'll put that in, hit 100% on it, or hit absorbance percentage on it, and then it subtracts off any absorbance due to acetone, so that we're left with just the absorbance due to the carotenoid pigments. Then what we're going to do with our spectrophotometer is spend some time, a few hours probably, looking at maybe every wavelength of light between 400 and 700 nanometers, because this portion of the electromagnetic spectrum is called photosynthetically active radiation, or what's called PAR. PAR is photosynthetically active radiation. This is the radiation that we know is important for photosynthesis. We're about to get to why that is here in a second, but I'll give away the ending. This portion of the spectrum is important in driving photosynthesis. As an aside, 400 to 700 nanometers is basically visible light. So the portion of the electromagnetic spectrum that we can see and perceive is the same portion of the electromagnetic spectrum that drives photosynthesis. So one of the things we might ask is it a coincidence here that plants and people are using the same wavelengths of light? People are using it to see with, right, to perceive things, and plants are using it to make carbon-based molecules. And the answer is no, it's not an accident that this is actually the portion of the spectrum that represents most of the light that we get down here on the planet. The sun gives off gamma and X-rays and UV, but most of that is blocked by the ozone layer, thank goodness, because it's such high energy stuff it would cook us. And the wavelengths above UVB get through the ozone layer, but this portion of the spectrum, the 4 to 700, is the most abundant light there is, given off by our sun, and then above 700, the amount of light tapers off in that portion of the spectrum. Okay. Plus, this is lower energy light, probably not enough energy to drive photosynthesis anyway. Okay? So, back to our spectrophotometer. I'm going to set the dial to 400 nanometers, blank it, take a reading, and I'm going to plot that. And if I started with the carotenoids, that would be my data point right there. Then I would go to 401, blank it, take a spec reading of my sample, and continue this, and I get something that looks like this. Okay? That's the carotenoid pigments here. So this tells us that this portion of the spectrum is being absorbed by the carotenoids. Specifically, the blues and greens are being absorbed, and the red, oranges, and yellows are being reflected. So the reason the carotenoid pigments look red, orange, or yellow to us is because that's the kind of light that they reflect. They absorb other parts of the spectrum. Let's do the same thing with chlorophyll A. Chlorophyll A does this. High absorbance in this blue portion of the spectrum, lousy absorbance, lots of reflection in the green portion of the spectrum, and then here, absorbance into that red, orange, and yellow portion, specifically the red portion of the spectrum. So if I'm absorbing blue and red and reflecting off green, chlorophyll A appears green to us. Chlorophyll B, same thing, except its absorbance is closer to the blue rather than the violet portion of the spectrum, and absorbance is a little more toward the orange than the red, but it would still give off green light because that's what's being reflected. So in my chart here, the appearance of all the carotenoids together is this color, chlorophyll B is that color and chlorophyll A is that color, based on the light that's being reflected and the light that's being absorbed. Doing okay? All right, so we know that in this leaf there are lots of pigments. There are some red, oranges, and yellows, and there are two different types of green pigment, chlorophyll A and chlorophyll B. Okay? Incidentally, in that spinach leaf there's lots of red, orange, and yellow pigment, but you don't see it because the bulk of the pigment that's found in a leaf is chlorophyll. Okay? Lots of chlorophyll A and B, most abundant pigments in a leaf, so that the little bits of red, oranges, and yellows in the leaf aren't seen because there's so much green that it's like a kid took a green crayon and covered up the reds and oranges. We notice these in the fall because the carotenoid pigments are fairly cheap. Chlorophyll is expensive to make, so the plant, before it drops its leaves, will pull the chlorophyll back into the plant, leaving behind the carotenoid pigments, and then those get dropped when the leaf is dropped. Okay? All right, these are the pigments. Now the question is, are these pigments involved in photosynthesis? So to test that, we're going to use a slightly different approach than the one we used here. This approach is called the absorption spectrum. It's giving us a picture of what wavelengths were absorbed by the pigments and what wavelengths are reflected. Now what we're going to try to do is figure out if these different wavelengths of light actually drive photosynthesis. So what I'm going to do is take a little alga here, okay, and I'm going to take this alga and I'm going to put it into my little cuvette. And I'm going to put it in a spectrophotometer. Okay? Still has that plate behind it. But this kind of spectrophotometer differs a little bit in that it also has a probe. And that probe that's here is an oxygen probe. And what it does is it senses the concentration of oxygen that occurs within this liquid. Does that make sense? The idea is the plant gives off oxygen as a